the very last thing that I wanted to talk about for section 13.2 is how do we know specifically what colors certain things are absorbing. So when we're talking about absorption of visible colors, we use something called UV/visible spectrometry. It can detect wavelengths in the UV to visible range. And the way that it works is that we start out with a light bulb that emits all the colors in the spectrum. So we have a light bulb here. Okay, it emits all the colors of the spectrum, and those colors pass through a transparent container known as a cuvette that contains whatever sample we're interested in analyzing. After it passes through that cuvette, we basically have a screen that can observe, so I'm going to draw an eye, that can observe specifically what colors are passing through my sample. Okay, so again, going into the sample, because it's white light, we have all the colors of the visible spectrum. Having trouble drawing arrows. And then coming out, we have certain colors that are absorbed by our sample, and so we only see certain colors. So let's just try to show that this is a rainbow. So again, white light is a combination of a bunch of different colors. Okay, and then coming out, perhaps we might not have green, for example. So we have all the other colors coming out, but we don't have green. Okay, and so we know that this sample absorbed some light. In this example, arbitrarily, I chose green. So let's talk about what that means. So before we put our light through our sample, so this is the color wheel of before, we have all of the colors. So let's color that in. So we had red, green, yellow, yellow, purple, blue, and I need an orange. Let's choose this one. Okay, so before we had all of the colors appearing in our light, and so to our eyes it appears white. After, we've removed one of the colors. So we've removed the green color. Okay, and so what happens is, according to our eyes, now that we no longer see this green color, the sample actually appears red to us. So we've removed, the sample has removed, by absorbing green photons in order to provide enough energy for my D to D transitions to occur. And so the removal of green to our eyes makes the sample seem like it is red. So we can tell exactly what color is being removed by these metal ion complexes by looking at the exact opposite, at the complementary color of the color that we see. So if we're seeing red, we know that our complex absorbed the green wavelength of light. So removal of complementary color gives red. Okay, and you can do this for any color. So if we see yellow, what is really going on, if we see purple, what is really going on is that our metal ion complex is removing yellow. If we see blue, what is really going on is that our metal complex is removing the orange wavelengths of light. So what we see is a complementary color of the wavelength of light that is needed for that D to D transition, for that stabilized to destabilized transition of the electrons.